So today we'll finish the homology modeling by going to the actual modeling step, how it occurs. Okay. So yesterday we did what I did in the lecture, that we did the blast. We found that how I have shown you how to do the blast, and also I have sent you some video links apart from my lecture yesterday, where from the video links you can also see how the blast, all these things is done. So from blast I have told you that we choose our template sequences from the blast, which has the more identity. With the more identity we choose the template sequences. Okay, and with the template sequences we run our homology modeling. Okay, so if I write a flowchart of this first, how this is done, I am writing the flowchart here first. There is the first one is having a query sequence. Query sequence means whose sequence you are going to model. Sequence that is for modeling. Okay, this you have to do the modeling. Now how then what do you do? After we have the query sequence, we proceed to the next step, that is we choose the template sequences, right? Choose the template sequences. Oh, and also I have sent you some Ramachandran plot and all these plots video links. Apart, I have not yet uploaded my video links in YouTube because there are some network issues yesterday, so I couldn't upload. So maybe today I'll upload my yesterday lectures. Apart from my lecture also, I have sent you some video links of Ramachandran plot, and from the Ramachandran plot you can explicitly understand how Ramachandran plot is interpreted and how it is done. Since you have done the yesterday's class, now I think you will feel easy to understand all these Ramachandran plots and blast all these things. Okay, so please go through those video links and the notes I have sent you, and the Google Classroom class code I will share today. So accept the Google Classroom class code and join the Google Classroom classes where I posted all the video lectures and the notes, everything. In the yesterday's lecture, yesterday's videos lecture, I have already put in my Google Classroom. So if you go to Google Classroom, you can find out those. But okay, I will share the class code today, and I'll also try to upload in YouTube channel, my YouTube channel. Okay, so choose the template sequences. Why? How you can choose the template sequences? By running blast, seeing the percent identity. Okay, now you run modeling. After that you run homology modeling. To run homology modeling, and homology modeling by different programs. Okay, and then you run the validation. You run the validation. In validation, we'll have three specific validation methods. One is Ramachandran plot. Second one, erect plot. And the third one, RMSD calculation or RR plot, residue to residue plot. Okay, this is the basic flowchart that we want to follow. Okay, if the validation is okay, then we can accept. If this validation okay, okay, if all three validation are okay, okay, then accept the model, then go for further studies, go for further studies. Like what if you want to do docking, or first you want to have the binding domains, then you go for the docking, and from docking you can do the structure based drug design, or then using that protein you can do the virtual screening of a library of ligands, you can do the de novo ligand design. Okay, so you can go for the further studies. This is the basic flowchart. Now we have seen the blast, how it is done, and how the percentage identity of proteins are seen, and how the templates are chosen. Now the homology modeling part. This homology modeling part, there are different softwares. The one is like Swiss model, and the other is Modular. Okay, one is Swiss model and other is Modular. I am showing you. I am going to again a screen share. Okay, the first I'm going to internet to show you. There is, you go to Google, fine. You go to Google, say I'm going to for Swiss model. You just type Swiss model. Okay, Swiss model is a modeling server by XPASI. XPASI is the that European Molecular Biologist Union protein analysis server. So there they have also given that molecular modeling options. The Swiss model. Okay, you go to Swiss model. Now you go to start modeling. So what you need to paste here, that is your target sequence. What you need to paste here, that is your target sequence. Okay. For example, I'll give you one realistic example. Like say, I have done molecular modeling of one protein that is of aldose reductase activity. Aldose reductase means is a reductase kind of enzyme which converts aldose to alcohol, means carbonyl groups to alcohol. Okay, that is a kind of reductase enzyme. I have done that carbonyl reductase molecular modeling. So what I need to do first? I need to go to NCBI. If you want to do by Swiss model, just go to NCBI. Okay, then you go to protein. Okay, you go to, I have shown you yesterday, protein is, you choose protein from here. It is already chosen. So say I am doing carbonyl reductase from Candida glabrata CBS 138. Say this is the one enzyme. Say this is one enzyme. Carbonyl reductase Candida glabrata is CBS 138. So once you search, okay, you will be getting lots of protein. Okay, so like that you can search any protein. Like that you can search any protein. Say if for yesterday, say I searched superoxide dismutase homo sapiens. I can search this will be superoxide dismutase homo sapiens. Yeah, so you got the proteins. Okay, this is a full protein that I analyzed yesterday. I did the blast. So for Swiss model, then you go to faster format. For Swiss model, you don't need to do the blast. They will do for you. for Swiss model, you don't need to do the blast and you don't need to find out the more percentage identity proteins, template proteins, that is the PDB proteins. You don't need to do, they will do for you. Okay, so what you need to do, you just copy the sequence from here, just copy, and you paste the sequence here. Okay, you paste the sequence here, and then you just go for build model. You just go for build model. Okay, automatically you see they have taken it. They have taken it. They have taken this target. Target means that our query. Queries means this is the target one. So then you just hit build model. Once you hit the build model, okay. Once you hit the build model, okay, they are running the project for you. And once they finish the project, the model protein will come here with different informations. Here what they do first, they will search the template. You see here template button is running. That means they are now running the template search for you. What we did in blast, they are now, they will do the template search for you by the percentage identity match and all of the everything. Then you will do the sequence alignment to find how much identity is there. Okay, and based on the templates they will build a model. This may take some time. Okay, like for let's say few minutes to few hours this may take, so you have to wait patiently. Sometimes you can give your email ID and all these things, so once their model is finished they will revert back to you through your mail that this model is ready, and these are some the validation data, some of the validation data they also give to you. This is the Swiss model. This is a very, you see, template they have already done. If I click here, you can find that what kind of templates you see. This kind of, they have done the blast analysis and all these things. So they have found these templates, and from these templates they will choose certain templates based on this. You see the person identity they have got. So then person identity they have got like this. So based on this, they will choose the templates and they will do the modeling job for you. This is one. Second thing is that if you want to run modeler, what I prefer, then you go to internet. Okay, then you go to internet and you do the modeler. You type modeler, and this is the modeler, the Andres Sally lab. The Andres Sally is the pioneer of this work, modeling the modular software he has coined. He has designed this. So from his web page you can download this. Okay, actually it is commercially available software, but if you are from educational institution, you can easily download this. Only you need to log in from your educational institution ID. You have to prove that you are from education institution and you are doing for non-profit use. So you have to use for your educational institution ID, and you have to give your educational institution details. So you just log in and give all these details. So they will send you a code in your email, in your educational ID or whatever ID you give. They will send you the email in that. Only thing you have to prove that you are from an educational institution. That is your educational credentials. You have to input, and they will verify this, and then they will send you the code. Using that code, you can download and install the modular in your software. Okay, they do all free for educational institutions. Okay, I have downloaded like this. So this is modular. You can download. After you run the modular, download the modular. There is some tutorial there. Actually, this is a Python-based program. Python is a computer language. Python-based program. It is run in DOS prompt, MS-DOS prompt. Okay. In Windows, you just go to MS-DOS prompt, and there are 5 to 6 script files. In the script files, they will give you the files only based on your target sequence. That means based on your query sequence and based on your template sequence, you have to modify the script files. The main syntax is there. Only you have to modify as per your query sequence, as per your target sequence. Once you modify, just save. Okay, and then you go to MS-DOS prompt. You run the script files with command. This command ends with .py. .py. The command ends with .py. The .py means Python. That means Python based script. Okay. So after you finish this modular running, the 5 to 6 script files, basically there are 5 script files which will make the model for you, which will make, after you run the initial 5 script files, the molecular modeling will be over and you will get the protein model. The sixth script file actually will refine the model for you. Refine means the 5 first build models may be containing some errors. So the sixth file will refine those errors and will more polish the model, will make the model more accurate. Okay, this is modeling. So it is about molecular modeler or modeler. Just download and do this. Okay, so I am again going back to my whiteboard. Actually, here, if you use modular, and I'm writing here, if you use modular, basically 5 script files are there based on Python language. Okay. Based on Python language. This makes the homology model of protein. Okay, and the sixth one makes refinement of the model. Okay, that means make the model more accurate, makes the model more accurate. Okay, if we have separate practice session, I can show you how the modular works, because I have already done and I have one folder already containing this. But right now, this is for tutorial, so learn this first. So the sixth script file makes the model more accurate. Now I will show you some practical examples. So I am going to here, screen share. Now this is a folder. Where I have done the molecular modeling. Okay, which molecular modeling? Actually, there was one protein I worked with that I told you, that carbonyl reductase, the aldoketo reductase from Candida glabrata. Okay, so I found that I needed to do the homology modeling of this, because I wanted to do docking on this protein. But when I went for docking on this protein, I found that there is no homology model, there is no PDB protein structure available for this one. So if there is no PDB structure, there was no structure allotted in any database for this protein in the various in the universal protein databases. So I had to do homology modeling for this. Okay, I tried different homology modeling for this, but in this session I cannot describe them all. But I'm showing you some key features. What I've already shown you one Swiss model. I have also tried the Swiss model for that, and I also had the data. I tried both modular and Swiss model for this to predict its structure. Okay, so first I found that this CR protein has four. When I run for template search, I found 4 templates are there. That is your this one. You see the code YDR541C, YGL157W, YOL151W, and there was another one. These 4 proteins were posing high sequence identity with that. Were posing high sequence identity with my CR protein. So, but the problem was that these proteins were also not reported in PDB. That means these protein I didn't find the structures. So for these proteins, there is no PDB structure available. So each of these protein I modeled. Each of these protein I constructed structure by molecular modeling. How? Actually, when I ran one of these proteins, okay, when I ran these proteins, I found 4 other proteins. I found 4 other proteins for each of the protein. When I ran blast, I found 4 other proteins which can be taken as template and reported in PDB. That means if I run this protein in blast, I found 4 proteins. If I run this blast, I found 4 proteins. If I run this blast, I find 4 proteins. All these 4 proteins are reported in PDB, and all 4 proteins are almost same. So using those 4 proteins, I modeled each of these. Using these 4 proteins, I modeled each of these. And after I model each of these, now I have all these 14 structures. And then using these all protein structures, I basically did the modeling. Of my target CR protein. These 1 UJM, 111P, 1Y1P, 2C29, and 2RH. These 4 proteins was used to template these 3 proteins. These 3 proteins I have to take as template for modeling my target protein, because these 3 proteins are highly identical protein with my target protein. Okay, so first I did the modeling of my template proteins, and using those template proteins I ran my modeling of my target CR protein. And after I ran the modeling for my target CR protein, I show you one that I got the model. Here, this is the one of the model I got for my target protein. Okay, this will open in one software called Discover Studio, because not all software can read your protein, like Discover Studio or ECSF Chimera or PyMol or Rashmol. You have to install this software to read the protein. So here, this is the protein I modeled. This is the protein I modeled. You see, this is not what I got, this is not obtained from PDB. This is from model. Okay, so this was the output from modeler. So after that, the first thing you need to do, you go for the modeling. Is when it is over, basically you need to do the model validation, right? You need to do the model validation. So first I am going to show you something in whiteboard. This is the model validation steps: Ramachandran plot, ERAT plot, RMSD calculation. One thing before doing that, one thing I forgot to mention you, 2 things you need to check. One is called dope. One is called dope score, and other is called GA34A1 score. Dope score and GA34A1 score are given by modeler. This is given by modeler. What is dope score? The dope score is basically, the full form is discretely obtained potential energy. Discretely obtained potential energy. That means if in modeler, what they do for you, they give you several protein model like this. That means several protein conformations like this. Okay, in the script file, you get the several protein conformations. Like I forgot, approximately 4 to 5 conformations they give you. So Which conformation to choose? You have to choose the conformation having lowest potential energy. The conformation you have to choose lowest potential energy, because you already studied the molecular dynamics. The lowest potential energy one has the most stable conformation. We have also studied in QSR that every compound is stable at its lowest potential energy conformation. So protein is also having the lowest potential energy conformation. So you have to choose the protein model with lowest potential energy conformation. So you have to look to the dope score and you have to choose the lowest dope score one conformation from that. This is one, and this is the GA34A1 score. GA34A1 score is the score where what software does. Software takes software takes your template protein. Okay, software takes your template protein and overlaps on the model protein. Software takes your template protein and overlaps on the model protein. Then they run a residue to residue distance calculation. They run residue to residue distance calculation. That means the residue 1 to residue 1, residue 2 here to residue 2, residue 3 here to residue 3, so residue 2 residue distance calculation. And from this distance calculation, together with the potential energy check, residue to residue distance matrix calculation, then potential energy check, and there is one more function combining all these three. They give you the GA34A1 function. The GA34A1 function basically depends on the residue to residue plot. If GA341, this one, is near to 0, then it is rejectable. If it is more near to 1, that is better. If it is nearer to 1, this is better. If it is near to 0, this is bad. So if it is closer to 0, this is rejectable. If it is more nearer to 1, that is more acceptable. Usually for GA341 plot, we take 0.7 or higher values. 0.7 or higher values as acceptable. Okay, 0.7 or higher values acceptable. Sometimes 0.6 or something is also taken. But if your protein conformation, if any of your protein conformation has the GA34A1 score more than 0.7, then it is highly acceptable. Okay, so you first look at your protein conformation to see which one has the GA34A1 score more than 0.7 or closer to 0.7. You first choose that one. Say this one is more closer to that acceptable GA341, then this one, you rank like this. Then you see the dope score. So say this one, the dope score, say this, for this one the dope score is minimum, and this has also, like, it is also second rank in GA341 scoring. So easily we can take this one as our final model protein. So easily we can take this one as a final model protein, because this is second ranked in GA341, so is GA341 score is 0.6. So we can take, and dope score is low, so it is the most stable conformation. So we can take this. So after that, we have the model in hand, which is built running the templates, choosing the base template sequences, and then running the molecular modeling or homology modeling, and then having the good potential energy and good GA341 score. Okay, this is GA341 score is good because residue to residue plot. This is also called Z plot. The Z plot of this residue to residue plot is also good. Okay, so after we take the model, we have the model. Now we have to go for validation of the model to see if they're okay or not. So first we need to see the Ramachandran plot. Yesterday we talked about lot in Ramachandran plot, and I think you have also seen a lot from the videos of the about Ramachandran plot. So how the Ramachandran plot is done is done like this here. Okay, here this is the Ramachandran plot of for my model protein. You see here. Where from you do the Ramachandran plot? You can easily do Ramachandran plot from here. You go, different servers are there. The best server is Procheck. You just go to type Procheck server or Procheck. Okay, you Procheck homepage. You go to Procheck homepage. Okay, and if you go to project homepage, you will find this. Okay, you will find this. You can download this also. You can download this also, and if you don't want to download this, then you can go to here, project here. Okay, here you need not to download. Okay, this is the server from NCBI UCLA .edu, University of California .edu. They have formulated this kind of pages. So you just upload your structure file here. You have the model structure, the PDB structure. The protein structure always stored in PDB, protein data bank format. So you just choose file. You just choose file from here, upload here, and just run. Once you run, you will have the something, the plot like this. Now you know the Ramachandran plot now. You know the Ramachandran plot now. So you just see that if your most of the residues in your protein lie in the core region or not, more favored region or not. The red one is the more favored region, and yellow one is the less favored region. I see that some erroneous region. Yellow one is acceptable, but not that good as red. They have some allowed regions, but core regions are red. So you see from my model that the black dots are the residues. So you see from my model that most of my residues lies in the core region. You see, most of my residues lies in the core region. Some residues are non-lying in the core region, but still they lie in the acceptable region. Very few residues here on the white portion. Very few residues are here which are outliers. That means not lie in the acceptable region. Very few regions are outliers. That means they don't lie in the core region. They don't lie in the least favor means some less favor region, but their number of residues are very less. So overall, you can see that more than 97 to 98% my residues of the models are good. They are within the Ramachandran plot, and most of the residues are in beta conformation, because this is the beta. You can understand this is the beta. This one is anti-parallel region, and this is the parallel region. Okay, and this is the alpha R, right-handed alpha helix. So most of my residues are belong to either right-handed alpha helix conformation or beta sheet conformation. So my model is a mixture of both. My model is a mixture of both. That means right-handed alpha helix and beta sheet. And in beta sheet, they have both anti-parallel and parallel beta sheet. Okay. So once you do the Ramachandran plot, then there is another thing I told you, that is you have to do the ERAT plot. Okay. So here ERAT is given. ProCheck-in also the ERAT is given. The ERAT plot. You just upload file here and you can do the ERAT plot. Okay. Also, there are, if you directly launch the ERAT plot from typing in Google, if you just type in Google, they will send you. So what the ERAT plot will look like after you do the ERAT plot, you will see something like this. So there is 95% confidence interval. This is the residue number. So you have 220. You have 220 residues. So each residue they will do the ERAT plot. The main fun of ERAT plot is that in the program they will set the confidence limit 95% and then 99%. The main funda is that most of your residues should lie between under 99% or 95%. Basically, it is better, that is, if most of your residues lies under 95% confidence interval. That means your data is 95% believable. That means your residue plot is 95% believable. If it goes beyond that, goes beyond 95% or 99%, that means it is going beyond your confidence interval. That means they are going beyond your 95% trustable limit. So those residues basically cannot be taken. So you see, this is the first one. One model was developed. They ran first the ERAT plot. They got several residues more than 99% interval. You see, more than 99% interval, even 95%. So they again refined the model. Like I told you, the modeler, the 6 program is there, which can refine the model, 6 Python script to refine the model. And in the refined model, you see most of the residues are coming within the 95% plot. And these one also, they are also coming almost in the range of 95%, yet they are touching 100%. That means they are well beyond the 95% confidence interval limit. They are not trustable, but now they are becoming trustable. Okay, so ERAT plot will give you how many residues are within your acceptable limit and how many residues are not. And if those residues which are not within the acceptable limit, you can run the refinement and make them happen within the trustable limit. So this is ERAT plot. And the third one is the RMSD one. The third one is the RMSD one. RMSD roots means square deviation. Okay. I'm going here. The RMSD one. RMSD is nothing. It is just like residue to residue plot. There are certain programs like PyMol or something like that where you do the RMSD calculation. RMSD, what you will do, they will first take one of your template and then they take one of your target, then they overlap and calculate the residue to residue plot. How much distance is there? Usually RMSD calculation, if the residue to residue distance is less than 2 angstrom, they are acceptable.